Welcome everyone. Good morning for this hands-on session. We're going to introduce you with a few tools based on the NEURON simulation environment that are available in the cellular level simulation, interactive workflows and use cases that Michele briefly introduced a few minutes ago. Before starting with the tools, I would like to share with you the idea that Professor Biale introduced early this morning on the community work that we intend to do in the EBRAINS research infrastructure framework. And this community is based on a platform which is called Collaboratory. So the Collaboratory is accessible from the EBRAINS main website in the services panel, and here down here you have a link which is a direct link to the community. So if I click to community, there's another panel with a brief description of the Collaboratory, which is version 2 of the Collaboratory developed in the framework of the Human Brain Project. And here you have a dedicated page. So you can learn how to browse the Collaboratory, access to legacy Collaboratory, and so on and so forth. And down here, you have a direct link. So you click on Browse the Collaboratory, you are provided with the main page. You have a quick tool that guides you through the main features of the Collaboratory. You can end the tool whenever you want. And then, as you can see, I'm not logged into the platform, but I can already see a few things. So a few documents, a few links. Why are we introduced in the Collaboratory? Because the work that we are doing at the cellular level simulation, let's say work, that we are carrying on, is based on one of the platform that is tightly integrated with this Collaboratory, which is the Jupyter Lab platform. And Jupyter Lab means drive, means documents, means basically running code. So the Collaboratory gives you all these opportunity, as we briefly saw this morning. Now that I'm in the main page, if I click Collabs here, you can see that I can, yes, basically every time you enter a page that you have not visited, you are provided with a reminder for the tour. So for the quick tutorial that you can have in the page itself. So as you can see, there are a few Collaboratories that are already available here and that are public. So you don't need an EBRAINS account in order to see these collabs. And these collabs are organized as wiki pages. So you have a main page, which is shown in the main framework here. And then you have subpages, the installation of the software, image pre-processing, and so on and so forth. Then you can always go back here to the collab and search for some collab, for example, document. You see that here you have a few collabs that are about that contains the word document inside the title or the description. For example, the HPP image service user guide. So you open these, and then again you can explore the documentation. Okay, so we go back to the main page. But now you have a direct link to the documentation. You have a direct link to the support, which allows you to takes you back to the EBRAINS page for writing an email if you have any bug, any question, if you want to give your feedback, and so on. And then you have also a direct link to the forum where you can ask questions and also chat with colleagues for on specific topics. Now, I believe most of you have already registered to EBRAINS. So if you, from the main Collaboratory page, if you enter, if you register, if you log in the EBRAINS portal, you will see something more. So you will have the opportunity to do some more operations inside the Collaboratory portal. So for example, if I go back to the collab link here, you can see that on the left, this left menu, I can filter the collabs that I'm looking for by my role. So, for example, if I search collabs that are that contain the test word inside them, and then I only want to see the collab that I'm an editor of, then I will be filtered out with all this collab. If I uncheck also this one, you can see that there's no, I mean, there's a shorter list, of course. I can see the collab that I'm a viewer of, and so on and so forth. So basically, the collab are workspaces that you can use with your colleagues and collaborators for sharing data, sharing documents, and also give the correct rights to the people who are accessing the collab itself. But now, how do we create a collab? We create a collab by clicking this button, create collab. Then I will put my initial here's EBRO, for example, EBRAINS like this. You have a collab title, you have a collab name, you can insert a brief description here, and also you can choose whether this collab is private or public. Let's say we keep it private for the moment. So just a sample collab. Takes a few seconds. Now the collab is created. And as you can see, as I was mentioning before, the main layout is the one of a wiki page. So this is the main entry, my collab extended title. I can edit this, of course, so I can enter the all the text, for example, the text that I want, sub title. Okay, I can change the text here, and then save and view, and the document has been changed. I can also add new pages. When I select the create page, I have a few options here on the right side. I can add an article, a blank page, or a community app, which is one of the web applications that we are going to see in a few minutes, and will be visualized. If I add one of these, this will be visualized in the main in this main central frame. Just as an example, I will add an article page here. So main article, which is the title. I create this, and there's a template which is used for creating this article. And one of the nice thing of this document, so the article template, is that you have headings here. Okay, so you can create for Office Word documents, you can create headings, and then you will have a table of contents on the right side. Again, you can create another page, which will be a sub-page of this. So sub-page article, let's call it like this. I will create this page, save and view. And as you can see, this second page is now a sub-page of the main one. Okay, so so the wiki is a very powerful tool for sharing documents, Excel files, for example, and other let's say light files with your collaborators and partners. For large size data, this bucket functionality has been created. So buckets and drive both are used for putting some data inside there. But while the drive is mostly designed for Excel files or presentations or live documents, so things that have let's say some kbytes or megabytes size, the buckets is for larger size. So you just initialize your buckets. You can upload the file, for example. I can go, for example, here, take, I don't know, this file, 7 megabytes, start upload, and it is uploaded here. So the bucket gives you the functionality of having more space, but the access is not direct. For example, from Jupyter Notebooks that we are going to see in a few minutes, but you have to use APIs for using the bucket. Okay, while the drive, which has a similar functioning, again, also here I can upload the same file, for example, but this will be in a different location. This is directly accessed. Now, if you have larger file to be uploaded, please contact the support team in order for them to give you the right advices and suggestions depending on your tasks. Now, there are two other links on the left menu, which are the lab and the team. The team, as I was saying before, is for managing accesses to your collab. At the moment, there's only one user, which is me. I can add another user as administrator, or I can add a group as an administrator or units as administrator. Of course, groups and units are a collection of users, so to speak. But also I can give accesses as editors, so not administrators. Editors can change documents, but cannot really, insert other, do higher level operations like inserting new team member or give access to units. And then viewers. Viewers are users that can only view the documents in the collab. So for example, if I go to editor here and I add Carmen to this collab, I will see her name here. I click on Carmen, Lupascu. Now the user has been added, and in a few seconds, you can see that she's an editor of this collab now, and she can modify the content. Finally, the lab. The lab is directly linked to the Jupyter Lab domain of the EBRAINS. By default, I see the drive here of my collab, and you can see that there's only one file here, which is the one that I uploaded before. If I close this file, which is the latest file that I edited yesterday, I am provided with all this functionality of the launcher. So I can create a notebook here, like import OS. This is for Python scripts. And then we can write print os.list dir, for example. And here we have the content of this directory. Okay, I can also open a console, which is basically Linux console with basic functionalities. So you can use this, for example, for removing files or cloning Git repository with code and so on and so forth. Now, this lab and this drive sections have a dedicated domain, subdomain in eBrains, which are lab.ebrains.eu. And once you go there, you are provided basically with the same interface, but on a larger site, so larger window, and you can explore all your libraries, which are the folders from your collab. So now you're not stick to a single collab, you can explore. You can also do this in the other interface, but you can do this from the lab domain in a more comfortable way. And the same holds for the drive. So we also have a dedicated subdomain for the drive. And again, here, I have all the drives that are shared with me, no libraries shared with me. My libraries, I can open these. And again, I have some work that I created in this folder. Share with groups, again, all the folders from my collabs. Okay, so this is just a brief introduction to the collaboratory, because we are going to use the lab, and we're going to see how the lab works. So it is useful for you to know that you have this very powerful tool for sharing content and data with not only collaborators and partners, but also with the public, if you have some documents that you want to make public. Now, if you go back to the eBrains main page, as we briefly mentioned earlier this morning, we have many tools and services that are available here and are gathered and collected by topic. We are interested in the simulation one, and more specifically in the interactive workflows for cellular level modeling. Again, here you have a page with a brief description. You have seen this with Michele this morning, so you have the guidebook. So read the guidebook, as Michele was suggesting before, using the tools, and then you go to the direct link of the page. Now, in the main page, you have general information. You have a section with the objectives of the work that we are carrying on, and all the tools and services that we are designing and implementing. You have a direct link to the workflows and use cases that we are developing, which is actually what you are going to use to do some research work. Then we have a link to the monitor of the HPC systems. As we have seen this morning, most of the heavy work and jobs and processes are run on high performance computing systems, and some of the users, the partners, but you might be one of these users, have their own account on HPC systems. So you might want to see whether you have quota left on one of the systems, or what's the status of the system, if it's online or not, and so on and so forth. And then we have a light papers section, which links you to a page which is a simple HTML interface. So this is just a preview. If I click on explore, I go to the direct link, and this is where we collect all the scientific work that we are doing in the framework of EBRAINS and the cellular level simulation platform, with interesting links to not only general information, for example, on the paper themselves. I will open this one, for example. But in the framework of sharing data, sharing software, what we have seen this morning, we provide links to data. For example, this manuscript was about physiological variability of channel density in hippocampal CA1 pyramidal cells. And in order to write this paper, we needed to use morphologies, so reconstruction of neural morphologies. We needed to use electrophysiological traces as well. And also we developed a model and so on and so forth. So these light papers is a place where we put not only information on the paper itself, but also links for downloading the data. For example, this one, as you can see, the file has been downloaded. And also we link this data to the tools that we are developing on the platform. So we have developed a tool for a morphology viewer, which is this one, and we link this file on the morphology of a neuron to the viewer. So you can zoom in, zoom out, you can realign, and so on and so forth. Okay, and the same holds for the other data. For example, here we have electrophysiological data. We can download the data, or we can visualize the traces. So in this case, this is a file with many traces. I will take the last one, which is the one where the higher stimulus has been given, and you can see here that we have the spikes event. Okay, so we go back here to the portal. Finally, you have some information on who we are and the links for contacting us. Now, the core of this platform are the workflows and use cases. As you have seen, you have many topics, many families, if you want: trace analysis, the small circuit in silico experiment, the single cell in silico experiment, and so on and so forth. You have these small icons that Michele was briefly mentioning. If a tutorial is available, you click on these and you open the tutorial.
The first page of the Neuro feature extract shows a brief introduction to the tool.
So you hear the voice that explains how the tool has to be used, and you are guided basically through all the steps. And then you have other icons that tells you whether this is a beta version, it is for power user or not, and it is implemented as a web application or as a Jupyter notebook. Okay, so we'll go back to the web application section of this this page later, but now I think it's time for Carmen to start talking about the Jupyter Notebook. So I will stop sharing my screen and give the floor to Carmen.
Thank you, Luca. I will try sharing my screen now. As we know, our hands-on tutorial today will be based on the NEURON simulation environment, which is used for building and using computational models of neurons and networks of neurons. We will see later how to what we can find on neuron.yale.edu, and in the same time, you can use the NEURON book by Carnevale and Heinz from 2006. Also you can go to ModelDB in order to, you can run and download already published computational neuroscience models. And NEURON may be used together with Python, and it might be used in Jupyter Notebooks. So based on this, let's go to the website, neuron.yale.edu, because here we have some information on how to install NEURON. We have also a lot of the documentation, publications, courses, and resources. And if we go to documentation, I think Adi has his mic on, and can you switch it off? Okay. So if we go to scripting NEURON basics here, we can see how to start with a basic NEURON script in Python. And we will do the same in the EBRAINS platform. So if you already created a collab, like Luca described previously, I already created a Jupyter Notebook in a collab called VMC NEURON. So you can download it and upload it in your collab. And here you can see how to, first of all, install the NEURON version and how you can use it. So in the first cell, we will install the NEURON version. We will import it. And here we can see how to print our version, which is 8.0. And in the Python world, it's important to import the H model from module from NEURON. And the first basic thing that we will do, we will create a section called SOMA. And with the psection method, we can see what's inside this SOMA. So for example, there's a default morphology, the length, the diameter, and the number of segments, the radius, and so on. But we can access those properties like length and diameter and change it. And of course, we can insert also mechanisms inside this SOMA. So for example, we will insert default mechanism, which is the Hodgkin-Huxley sodium, potassium, and leakage channels. We will insert it into the SOMA. And we can see some methods from this mechanism. So we can see there are some conductances of the potassium, of the sodium, and leakage channels. And we can print, for example, the conductance of the potassium channel inside this Hodgkin-Huxley channel. So we can see that the default conductance is 0.036. Then we can insert a stimulation, an iClamp, and we can, of course, define the delay, the duration, and the amplitude of this stimulation. And let's see how our SOMA is changed now. So we can see that we have defined the point process, the enclamp. We have the density mechanism here, the Hodgkin-Huxley, and the morphology is with the length and diameter changed, and so on. So now let's set up the recording variables. So we'll need a vector to record the membrane potential and a vector to record the timestamp. And then using the std-run Hawk file, we will be able to run the simulation. But first, we have to initialize our simulation such that our cell has a resting membrane potential of -65 millivolt. And then we will continue our simulation until 100 milliseconds. So now our simulation was quick. We can plot the results and see what happened with the cell. So we have here the voltage recorded. And this is just a basic script for those of you that never used NEURON before. I hope you can run it yourself in your own collab. At the end, we'll have time to see if you have questions on this and if you were able to run it. Then, going back to my presentation, I'm going to introduce some use cases that are scientifically more complicated. So the first one describes a general fitting procedure to study subcellular models of transsynaptic signaling at inhibitory synapses. We have a publication here from 2016. Basically, the workflow started from some experimental road traces from the EBRAINS knowledge graph. And we defined a NEURON kinetic model, so a mod file. We'll see later how this was built. But basically, the user defined some names and initial values for the parameters to fit, some constraints for the parameters to fit, so min and max values, some exclusion rules, and some dependency rules. And after we clip and remove the baseline, an optimization procedure is done using the NEURON built-in praxis, so principal axis steepest descent method for minimizing the error. And we use the parallel implementation that uses the NEURON's parallel context. And this is done using HPC platforms. And at the end, the set of the fitting parameters were analyzed by the user. We'll see how later on. At the synaptic transmission model that was described here, and that was and the parameters of this transmission model were optimized, has a basic time course of the synaptic conductance change that was modeled using a typical double exponential with time rise and time decay, as in Zodix and Markram. But it was a more complicated model because involves a protein that is called gefrin that in humans is encoded by the GFN gene, and it is a component of the postsynaptic protein network of the inhibitory synapses. And it has a complex role in modulating the number of available postsynaptic receptors and the amount of the neurotransmitter that is released at each synapse. And in this kinetic scheme, the inhibitory current is elicited by synaptic activation, is modulated by some variables like the gefrin clusters, the neuroligin and neurexin clusters, the neurotransmitter molecules, the postsynaptic receptors, and of course the basic time course of the synaptic conductance. And here we have the differential equations that were used in order to describe this model and that were implemented in the mod file in NEURON. Okay, so here we have the mod file, and we'll see that it has some blocks like the neuron, the parameters, and the equation and the differential equations that I showed you before. And what actually happens if we have the individual synaptic events recorded from the voltage clamp experiments, if we would do it by hand, it will be very hard to find the combination of such a big number of parameters. So you see here that it's difficult to find the perfect match. But if we use the configuration file, so written as here, so we specify the number of traces, we specify the protocol, so the voltage clamp amplitude, the reversal potential, and the initial fitting parameters, some constraints and dependency rules, we will find using the optimization procedure the right parameters, and the match will be easier to find. Okay, so before going on, I'll let you see how to do this into the EBRAINS platform. So if we go to services like we've seen before, and go to simulation and to cellular level simulation, we can explore the workflows and use cases. And we go to trace analysis, to synaptic events fitting. And what happened is that the Jupyter Notebook will be cloned into a destination collab that we can choose. For example, I choose the VMC NEURON 2807. Okay, and so the Jupyter notebooks are cloned from GitHub to this collab. We see here the cloning procedure. Okay, so once this is done, the Jupyter Lab is opened, and we can run all the cells in this Jupyter Notebook. We have here, as Luca said, in our drive, all the files necessary to run this use case. So we can download a specific data set from the CSS storage, from the knowledge graph, and the traces are shown here. And we can select a default model or a local one, so we can upload our own mod file. We can specify the number of traces, the name of mod files, and all the parameters that we've seen before. But let's go on with the default mod file. And we have here the Neuroscience Gateway HPC platform. We will choose the demo version, so we can log in to this HPC platform, and we can specify the number of traces that we want to fit, the number of CPUs, the number of nodes, and the hold time, and we can run the simulation. And from time to time, we can check the status of this simulation. Once this simulation will be done, we have a second Jupyter Notebook that that we can run in order to analyze the results of the simulation. So another graphical user interface is loaded. Of course, you can see older simulation from Neuroscience Gateway, so we can log in again and see the simulations. if they are completed, if they're still running or still in the queue. Of course, once you get this graphical user interface, you can switch back to the code by clicking here on Show Code. And in the second tab, of course, you can refresh until all the simulations are displayed. But here in the second tab, which is called View Analysis, you can, of course, analyze your results. So first of all, we have a results table where all the parameters are displayed, the parameters that were optimized. We can box plot the results, so we can analyze the box plot, or run NEURON in order to show us the best fit of that trace. Okay. Now, if we go back to the presentation, we will see, of course, before going on, in the trace analysis, we can find also synaptic events fitting with user model and with user data. So you will have also the possibility to upload your data or upload your models and do the fitting with your own data and own models. The second use case that I would like to introduce is how to build your own single hippocampal cell model. So again, we'll have some experimental traces and we'll have a mathematical neuron model, and that will help us to optimize the parameters in order to get a similar traces to the trace to the experimental one. So this use case leverages BluePy-Opt, which is an open source software that has an optimization algorithm that is based on the features that are extracted and on some parallelization technique that we'll see later on. And here are some examples of experimental traces. So the experimental protocol is just a somatic current injection, so just step current stimulation protocol. And the electrophys feature extraction library that Luca will present also later on helps you to extract features from the experimental traces. So for example, spike event features like time to last spike, inverse second easy, and so on. Some spike shaped features like the action potential amplitude, the peak voltage, and so on. Some voltage features like the voltage base, the steady state voltage, and so on. You have here a website where you can further visualize all the available features. And the code for setting the parameters, the objective and the optimization algorithm is partitioned into separate models that we'll see now. So we have a checkpoints folder, a config folder, figures, mechanism, model, morphology, and an important Python neuron that will help us to do the whole optimization. So in the config folder, we will see that some JSON file are available. The configuration values are stored and read from this JavaScript object notation JSON files. The first one is the morph.json, where the name of the morphology, in this case an ASC file, is used. So it's a detailed morphology. Then in the same folder, we will have the features.json that were extracted by the FL. So just for each current step protocol, we'll have the features, mean and standard deviation. And this feature.json will be read by the Evaluator Python file in order to read the features. So The objectives that are calculated are for every feature value is just the mean, the experimental mean minus the feature divided by the standard deviation, the experimental standard deviation. And this approach normalizes all the objective scores to a common scale, allowing them to be combined regardless of units and weights specific objectives according to feature variability. So at the end, we'll get a figure like this, where we can see the standard deviation of the objectives for each feature, for each step protocol. Then one other JSON file in the configuration folder is the protocols.json. So the stimuli are defined. The stimuli are just neuron square pulse stimuli, and of course we have to specify the amplitude of the current pulse and the step delay, the step duration, and the total duration, which specify the start time, the length, and the total simulation time. And these again are read by the evaluator.py in order to fix the protocols of the model. At the end, in the config folder, we will find the parameters.json. So are the mechanisms that are used in this model. And there are some parameters that are marked as fixed, so are kept constant through the optimization. And the parameters that are optimized are the conductances of the ion channels. And the location of the parameters is based on section list names. So these sections are automatically assigned by NEURON to somatic, axonal, apical, and basal. And this is done when NEURON loads a morphology. At the end, of course, this parameters.json is read by the template Python file. And in the mechanisms folder, we will find the mod files, so the mechanisms. Once we create a cell template to optimize the parameters of this cell, we need to create a cell evaluator object. And this object needs to know which protocols to inject, which parameters to optimize, and how to compute a score, as we've seen before. Once we have a cell template and an evaluator for this cell, the deep optimization can be created and run. So the deep algorithm is an evolutionary algorithm and evolves a population through consecutive generations. And for each generation or iteration, a set of offspring individuals are generated from selected parents from the previous generation. And of course, we can specify the size of the offspring and the maximum number of generations. The algorithm keeps track of the Hall of Fame, is called, of individuals during evolution. So not only the best evolution, the best individual, is kept, but 10 of them. Population statistics can be recorded in a logbook, and the genealogy between individuals can be saved, analyzed, and visualized. And this is done by checkpointing. And the deep algorithm stores a so-called Python pickle file for any generation. Okay. So here you have the template that is created. This is a Hawk file, so it's a NEURON file. And you can use this template with the optimized parameters in order to run a model, as you will see further on, explained by Luca. So at the end, the analysis produces into the figures folder three main figures. So the traces that are generated by the best solution in the Hall of Fame, you'll see here for all the current steps. Of course, the best solution has objective values that are in the same range as the reference model. You'll see the objective scores for the best objective. Of course, the goal is to minimize these values. And you'll see also the evolution of the optimization. So you'll see the minimal, the maximal, and the average score found in the consecutive generations of the evolutionary algorithm. Now we can see how this works into EBRAINS platform. So again, if we go to services, simulation, interactive workflows for cellular level simulation, and we'll explore the use cases. We will find this use case under the single cell modeling. So we will choose build your own single hippocampal cell models. Of course, if you are not logged in, you can log in. And again, the notebooks will be cloned in the destination collab. Together with all the necessary files to run this Jupyter notebook. So again, the Jupyter lab is opened. Here you can see the code, you can run it. And some of the cells are immediately loaded and run, but there are, inside this use case, this is a part where a CSCS container is downloaded and it takes a while. So I will skip this part, waiting for the graphical user interface to be loaded. Okay. So once this graphical user interface is loaded, let's wait a little bit. Okay. So you can see here the graphical user interface. And you can see a list of morphologies. You can see also the morphology. You can see the features and the parameters. You can modify the parameters as you wish. So you can modify the initial parameters and save the new parameters. You can specify the maximum number of generation and offspring size. And I will choose the service account on Pitsdaint, which is a free service that we offer to users that are not having an account on Pitsdaint. We specify the number of nodes, the number of cores, and we create a zip file that is necessary to be sent to Pitsdaint. And then we run the simulation. And we will wait until the results are ready. So the job is still queued, but we will see what happens when the results are ready. So when the status becomes from queued to successful, the results from the HPC platform are moved into the drive of our collab. Okay, so and we can run another Jupyter notebook which does the analysis of the downloaded results. Okay, let's skip this part, which is again a very long part. And let's go to the graphical user interface. Okay, and let's analyze the results from the storage. You see here that a folder is created, results Pitsdaint service account. And these results will be displayed here. So this is the morphology that we used, and we can view the analysis. Of course, in the background, it uses NEURON in order to run the simulation again in the Jupyter Notebook with the optimized parameters. Let's wait for the analysis result. Okay. And we will see the free PDFs as we have seen in my presentation. Okay. Okay, so here are the PDFs displayed. Of course, it does not make so sense. The results are not the best ones because I used not so many features or not so many generations in order to get the best results. Of course, we can show code, hide code, and so on. The last use case that I would like to let you see is a plasticity use case that configure and test through an intuitive graphical user interface different synaptic plasticity models and protocols. So again, if we go to simulation, interactive cellular level modeling, and explore the use cases, we will find this use case under the single cell modeling tab, and it's just called synaptic plasticity. And again, this will be cloned in our collab. We can choose a model from the model list. So you can see based on the morphology and the synaptic traces, what's the best model to be chosen for your plasticity model. And again, Jupyter Lab is opened. And we'll have the possibility to run again all the cells in order to get this graphical user interface. And what we will see here are mainly two tabs in the graphical user interface. Let me show you the, okay. So the first tab is the config. So you can specify the plasticity model to use. So this one, you can specify the synaptic parameters. We can specify the location. So if we want to put the synapses only on dendrites, for example, we can specify the protocol, so the activation pattern. So if we want to use a frequency experimental protocol or a theta burst protocol, or we can specify some additional current stimulation or some voltage clamp parameters. And at the end, we can place the synapses defined like this. And we will see that we can visualize the morphology with the specified synapses. Okay, so you can see here the synapses. And we can go in the second tab of this graphical user interface to specify the recording location, the number of simulations to be run, and we can plot the results And of course, we can download the results for further analysis. We will see here the results. So first, I just specified the recording site. So we'll see just the trace recorded from the soma. Okay. Here, but if we specify also the synapses to record from, we will see an additional trace that is recorded here. Of course, you have to pay attention to the parameters, and the simulation have more sense, of course. One last thing that I would like to specify, going back to the EBRAINS use cases, of course, that so let's go again to services, to simulation, to interactive workflows. And let's explore again the use cases, because I forgot to say that in the single cell modeling, you have another use case similar to this one, which does the rebuild of an existing single hippocampal cell model. Okay, because the difference with the build your own hippocampal cell model is again that you can choose a model from this list, and the Jupyter notebook that is downloaded uses already this model, and the simulation is very similar to the build your own hippocampal use case. Okay, you go to your Colab and do the usual stuff. Okay, I think this is mainly all. If you have questions, we can answer now to the questions before going to the other use cases that are mainly web apps.
Okay, so we can go back to the portal itself and have a look at 2 or 3 web applications, maybe 4 for single cell modeling and small circuit modeling. Again, these are web applications, so these are thought for users that do not necessarily have a background in programming or Python, maybe biologists or any other background that do not give you any knowledge on programming that is required for building your own Jupyter notebooks. Even though, as Carmen showed, you can create Jupyter notebooks with a graphical user interface that do not require you to program any single line of code. So here again we have our 6 groups for the workflows and use cases, and I will first introduce this feature extraction web application. So this is not strictly related, at least at the first glance, to modeling, because this web application is about extracting electrophysiological features from data, or experimental data, or also simulated data. But our approach in modeling is to start from data and then model neural models on the basis of what the data told us. So we want to have a look at the data, then we want, with the optimizer that Carmen introduced, we want to optimize a model against this data, and then we want to run the simulation and make predictions or confirm some experimental results and so on and so forth. So this standalone neural feature extract application basically gives the opportunity to go through the feature extraction from a data set or upload your own data. We have a list of contributors here that provided us, both partners of HPP and external contributors that provided us with electrophysiological data, that we can select from this panel. So for example, if I click on the fields, the keywords that you can choose are the contributors, the species, so mouse or rats, for example, the structure, hippocampus, cortex, cerebellum, and so on, the region, CA1 of the hippocampus, the type of the cell, pyramidal neuron, interneuron, and so on, and the E-type. E-type means electrical type, so bursting accommodating, continuous accommodating, and so on and so forth. So if you select, this is exclusive. So if I click on this one, as you can see in bold, I have all the options, which is a unique choice in this case. While if I click, for example, on Thomson Lab at UCL, I can see that the data provided from this lab are on rat, recorded from rat, hippocampus, C1, interneuron, but then you have three options here, which are the CNAC, CAC, BAC. Okay, so for example, if I click on this one, then I click on apply, I will show the traces that have been made available. Okay, so these are the traces. Now we'll go back to this point in a second. First of all, I want to show you this panel, which is about inserting feature extraction parameters. You have a model here explaining what all these parameters are about, so I won't go into the details of this. But for example, one important parameter is this one, the threshold that we are using for detecting an action potential. So usually the -20 millivolt should be enough. For example, if I go here to this trace, I can see that all my action potential cross the -20 millivolt threshold, but you might have other traces for which this threshold is not indicated. So you can set these parameters and also other parameters in this panel. And this is applied to all the traces that you are going to select for feature extraction. Now, another parameter that you can set for individual files, so not for all the files and the traces selected, but for these specific traces shown in this plot, is the voltage correction. Now, it might happen that experimentalists hold voltage while recordings, for example, to 0 for some reason, for some experimental reason. If this is the case, you might want to reshift all the traces to the original value, which is the 0 minus the actual voltage that was recorded from the cell. Now, if you need to do this, you can do this through the voltage correction panel. For example, again, here you have the instruction, the voltage correction value inserted by the user is added to all the voltage values. So if I click on plus, I'm adding 30 millivolts, 35 millivolts, and so on and so forth. And as you can see, the axes are shifted in order for you to understand what you are doing basically there while changing this parameter. Okay, so again, let's say we were here, we put again to 0, and we want to select 1, 0.8, 0.6. Let's say we select the same stimulus amplitude here. These traces, actually you can select all of them, neither of them, or you can select a few and then invert selection from this interface. But usually, if you're interested in spiking activity, the highest the stimulus amplitude, the highest the number of spikes that you might find in the trace. So, for example, with the stimulus of -2 nanoampere, you have an hyperpolarization here, and there's no spike here. Okay, so we'll take this out from our extraction. And finally, I will select these two traces also, and I will go on with the extraction. Another option that you have is to upload your own files. Now, the files that you upload are integrated with what you have chosen already here. So you might take a few files from here, from the dataset, and then you might upload your own file. For example, if I browse my folders, I should have a couple of files here. For example, this one and this one. I open this one, and a few metadata are already have been found already, because if you want to upload an ADF file, you need to, which is specific format for electrophysiological recordings, you need to give a metadata. Everything is explained in the guidebook here, and also in this short information panel. Then I selected my data, I upload the data, and as you can see, my data are here. So again, also here, I can select this one and this one. Okay. Next step is to confirm your choice and select the features that you want to extract. Now, the features are divided into 3 categories, which are spike event features, spike shape features, voltage features. Spike event features there are a few of them. Some are very intuitive, like time to first spike is the time from the start of the stimulus to the maximum of the first peak, time to last spike, time to second spike, and so on. For all of these features, you have a tooltip that tells you briefly what this feature is about. For example, spike count, and that's it. Spike shape features, we can select, for example, the spike half width or the action potential depth, and voltage features, so some other, for example, the voltage base. Okay, now that we have chosen our features, we can go on. The library that we are using, and that Carmen was mentioning before, is the electrical physiological extraction library, which is called FL shortly, and it is linked in this page. So you have the read the docs page if you want to know more about the library, and you have the PDF file, which is usually updated to the latest version of the feature description. So we go back here. We have chosen our features here. Now we go to next, and the extraction process has started. It is already finished. We can download the results. We can see here that we have a zip file containing all the results, and okay, there should be here in the download. So here we have our results. We can extract the files here. A folder is created for basically all the results. You can see here, you can also extract the results in a dedicated folder, of course. You have the mean and standard deviation of the features that we have selected in a PDF file. Then you have the JSON files that are used for the optimization that Carmen was mentioning before, and that we are going to use in a few minutes with another web application. And then you have the PDF files with the stimulus and the traces that you have chosen, so that you can have a look of what actually signals you have been managing. Okay, so this is basically all for the neural feature extraction. You can go back to the home page and start over with the neural extraction. Now, a second web application that I would like to show is the single cell in silico experiment. So the single cell in silico experiments under current clamp is a web application that allows you to choose a model from a data set and run a simulation on that model. These are single cell models. Now we are updating the data set from which we are fetching these models. So at the moment, if you're going through this web application, just choose the model with 2017 or 2018, because the 2019 are being integrated but are not yet integrated. So I will select this model. I have a few metadata here, a few information. I know that this model as this morphology has been created by this researcher, has been created with these channels in the NEURON format, and so on and so forth. So I select the model, I continue, and the web application is opened with this model inside it. Now we have a left panel where the morphology is shown here. We can zoom in, zoom out. We have the soma here. We have the dendrites, the axon. We can also have a look at the dendrogram of this morphology. So zoom in, zoom out again. We can select a compartment and have more details on that compartment here. So the capacitance, the morphology, diameter, the GPath, the other parameters specific to this model. Then we can go back to the normal visualization and start with the simulation. Now, as you can see here on the right panel, we have the current placement for the current injection, which is the soma. But if you click here, you will change the current injection. For example, if I want to change from this apex 16 to apex 32, I select the dendrites here or the compartment here, for example, okay, dendrite 26, and then I click on place current injection, and the current injection will be moved to this compartment. Usually, to have a look at the functioning and the behavior of the model, we select the soma. So I place the current injection to the soma, and now I can deliver my simulation at this location. In order to set my simulation parameters, I will click on the simulation panel here, and you can see that we have a bunch of parameters again. Temperature, the initial voltage, we have an OD current, if there's anyone, T-stop, a delay, duration of the simulation. So here we have 800 milliseconds, and for example, the amplitude of the stimulus, 0.7, we can put it to 1.7, maybe it's a bit too much, 1 nanoampere, and that should be fine. So we are all set now. I'm gonna run the start simulation, and we're gonna see here in this panel on the right bottom part of the monitor, the activity that we are recording from the soma. So let's click on this. And as the simulation runs, we see the activity. As you can see, when there's a spike, the morphology turns to yellow, because this gives you the idea of the propagation of the signal and also the intensity. For example, when this spike, when a smaller spike is fired, you have a less intense yellow color. Otherwise, it's very intense and so on. Okay, so we have now the activity. We can zoom in if we want to see, for example, the shape of this action potential. We click twice and we zoom out from the plot. And also we can download the activity in a CSV file. So this is a CSV file that has can be opened, for example, with Excel. You have two columns. In the first column, you have time. On the second column, you have the membrane voltage. Okay, I close this and go back to the portal that you probably are starting to know better now. And the next web application that I'd like to show you is, again, about single cell in silico experiment, is about single cell modeling. It's the same family of the build your own single cell and rebuild an existing cell that Carmen showed you, but this integrates a few more web applications from this portal itself and guides you through the entire workflow of a single cell building and simulation. This is called the Hodgkin-Huxley Neuron Builder. So I click on the Hodgkin-Huxley Neuron Builder. Again, we have a point-and-click graphical user interface. We can upload the workflow if we have one already. Now I will show you how to save your own workflow, or we can initiate a new workflow. Also here we have a direct link to the guidebook in the through the information icon and through the video tutorial, which is again with the old with the old interface, but the function is the same. We're going to update all the video tutorials in the very next weeks. Now let's start with a new workflow. Again, here we have basically 3 steps. We want to produce some data, extract some features from electrophysiological data against which we want to optimize our model. We want to select a model that we want to optimize from a catalog, and then we want to run the optimization process through the BluePyopt software package on an HPC system. The extract feature about integration is done through the Neuro feature extract web app that we have just seen. So we have integrated the web app in a model window in this web application. I will not go to the details. You already know them. So I will just select a couple of traces here and go next. Then I will select a couple of features here and go next. And then the feature extraction is performed. One option that you have now in this integrated window is that you can save the results of this feature extraction to the Hodgkin-Huxley Neuron Builder workflow. So save to workflow. I go back to the main page, and as you can see, this bar is turned green. This means that these data are already available. The second step is choosing a model. So I click on choose from the model catalog. Again, here I have a number of models from which I can choose that I want to optimize. For example, this one with these channels. I click on this, and also this one is turned green. Now, for all the results, you can download them. Here, you have a zip file with all the results. You can download the model and have a look at the model again. And again, as Carmen showed you a few minutes ago, when you download the model, and which is this one, you have also all the mod files, all the configuration files, all the parameters, the morphology files that you have seen. So if you want to change something in this model, for example, the mechanisms, you can do this. You will change the file here, and you will upload your own model in the zip format. Okay, finally, we want to submit the optimization process on an HPC system. So I click here, and we have 3 options: the neuroscience gateway, the Carmen showed, the CSCS pit-stained server, and then the CSCS service account. I will spend a few more words on the service account, but first of all, I will submit the job. Now, what you have seen so far is public, so you don't need any account in order to perform these operations. But if I want to submit the optimization process to the HPC CSCS system via a project that I'm a member of, which is this one, ICH002, I need to log in, because there's an automatic mapping done between the EBRAINS platform and the HPC platforms related to the Human Brain Project that is done. And so with the token that I'm using on the EBRAINS website and platforms, I am recognized by the HPC system. So if I click on apply here, I will have this message: you need to be logged in to use this HPC system. So close here, cancel, and log in. You have the login icon here. I am already logged in another tab, so it will recognize that I'm who I am already. So this is you can put also a picture of you here. If I click here, also this my personal page is opened, and so now everything should work fine. Okay, so we going back to this. I think we have lost the features, but we will upload a couple of files in order to speed up with the optimization process. So now I'm here. I have again my features. I have my model, and I want to submit the job. So again, I choose this 002, apply. Now everything is ready. This button is has been enabled in order for the submission to take place, and I can click on it. Launching optimization on the HPC system. A few seconds, and now the optimization is launched. Okay. Now, before going to the part where we fetch the results, I want to show you this. We have a few buttons here that allows you to create a new workflow. So if I click on this, this will disappear and a new workflow will be created. We can save the workflow. So we can download the current workflow on a zip file. So you see it is almost transparent. This is done, and you see the notification window here where the zip file is being created. And we can also clone the use case, the workflow. Why? Because as you have seen, once I submit the optimization, this button is disabled because every workflow has its own ID, and we don't want to submit the same optimization with two different IDs. Okay, so if we can submit the same optimization with the same data, the same model, but this will be a different workflow. So we don't want any confusion to arise there. Okay, so if I click on clone, a new tab is open with all the data that I already set, but now the workflow ID is different. You see here, we have a 026 in the end, while 827 in the end, and this button is enabled. Okay, next step. Once I've submitted my optimization process, I'll wait for some time, and then I try to fetch the result. Again, here we can choose between the CSCS stain, the CSCS service account that we're going to see in a few seconds, and the MSG. I click on CSCS stained. It is fetching all the jobs that I submitted recently. It takes some time because I have a few, not many. Depending on how many jobs you have submitted, this will take longer. And this, as you can see, we have the job that I just submitted a couple of minutes ago, which is running on the HPC platform. So I will take this one. It says, so I submitted this couple of days ago, or maybe yesterday. I will click on download, and let's see and wait what's happening here. So if everything went well, if the optimization went well, and I was able to fetch some results, then I will be able to see my model. This takes just a couple of minutes, so I'm not going to show you upload the model to show this, because as you can see, because I mean through this window you can see what the system is actually doing. So it downloaded the optimized model. Now it is running some analysis in order to produce the PDF files and the analysis that Carmen showed you before. So the how basically the genetic algorithms behaved and how the model is behaving after the optimization. It is creating the zip file that is needed for running the simulation, and now everything seems to be ready for the simulation itself. So I click on Run Simulation here, and a new window, model window, is opened with the same web application that we have seen a few minutes ago. So the blue NAS, the NEURON as a service. Same interface. Probably this is a different morphology. Simulation again, same thing. So I click on Run the simulation, And again, I can see my simulation here. As you can see, the output is not the same that we have seen with the other model. So of course, this is another model. So we are also implementing a functionality in order to register these into the eBRAINS model catalog, which will be ready in a few days. And the main point that I would like to stress here is that we are building web applications that try to use other web applications and other tools and other services that are already ready. So here we are using the feature extraction, so the neural feature extraction web application. We are using the HPC systems. We are using the NEURON as a service, so the single cell simulation environment, and we are gluing everything together so that the community can use all these tools at ease, I would say. Now, I would like to spend a few words on the service account that we have briefly seen when we submitted the job. So I will clone also this workflow, and again, I will click on this. Now, in this interface, as you can see, I'm not required for any project because the service account allows you to submit a job on HPC system without having an account on that HPC system. Let me change my my screen and give you a brief introduction to the service account. So full screen. So on the left side here, you can see a typical interface of a Jupyter notebook where the service account is provided to the users. This is a user with an HPC account, and this is a user on here in this column without the HPC account. Usually, to submit the job to Unicor, for example, in Germany, or to Daint in Switzerland, or to NSG, you need your own account. And the user, for example, a student without any account on these systems cannot submit the job. But with the service account, it can. It can because it only needs to register to the eBRAINS, and then we will take care of submitting the job on his behalf. What the system is doing is basically recording the ID of the user in order to know which kind of job he or she has submitted, the quarter that is using, the system that is submitting the job to, and so on. So we have created a server running on Cineca on a virtual machine where we created a small database that takes keep tracks of the user, the project, the quotas, the jobs, and so on and so forth. And then this database talks to CS Daint, in this case, to NSG, or to other HPC systems in order to run simulations, data analysis, and so on. The service account is documented here. And here we have a basically workflow of how it works. So the user log in to HP or eBRAINS in this case and request the service. So the user token is verified onto the OIDC system of eBRAINS and HPC, HPP. If it is verified, then this information is sent back. The user is authorized in this case, so the job can be submitted. And once the job is finished, the results are fetched back to the database and then finally to the user. Okay, this is an example of the database and the operation that you can perform. Submit job, fetch results, delete job, and so on. Now, going back to our web application, on top of this CS Daint with my own account, I can choose the service account. I can apply here. And again, this is ready for being submitted. Launch optimization, submission ended without errors. Now, if I want to fetch the results from the service account, you can see that I have a different list of jobs because these are the jobs that I have submitted through the service account and not through my own accounts here. And the one that I just submitted is queued. Okay, this is all for the Hodgkin Axis Neuron Builder. And there's another web application that I would like to show you. And this is called the Small Secret in Silico Experiments. So I click on the main tab here, and I click on the hippocampus. You have these both for the somatosensory cortex or for the hippocampus CA1 region, but the functioning is basically the same. So here I can see my region, and every dot of this figure are my neurons. I can zoom in or zoom out. Okay, I can shift, I can rotate, and so on. As you can see, if I keep on zooming in, I have a clearer idea. I mean, I can access more easily the cells in this interface. I can also increase the diameter of the spheres that I use for rendering the cells. And if I put the cursor upon this cell, for example, on the bottom left part, you can see a few information. The ID of the cell, the E-type, the electrical type, the layer, the morphology, the morphological type, the region, and so on. The colors are given by filtering different features of the cells. For example, the layer, and here we have 4 layers. I can filter by E-type, and here I can see that I have CA in orange, I have CAC in green, and so on. I can filter by morphological class, which are interneuron or pyramidal cell, and I only have 2 colors here. Then the morphological type, I have many, mostly depending on the region where these cells have been found, and so on and so forth. Now, I want to create a small secret, as the name of the web application says, with these cells. So what I can do is just to click on one of these cells, and this will be added to my cell group for simulation. I can click on another one, and I will have 2 cells here. But now I might want to filter the cells by some criteria. For example, connectivity criteria. I might want to take all the afferent of this first cell that I select, right? So in this case, I click on afferent here. I click on 34793 here. This will filter out all the cells that are not afferent of this neuron. Okay. I click on Add Filter, and as you can see, the number of cells is very reduced. I can push this further and set another filter. For example, efferent of the same cell. So if I click on here, we have afferents and efferents of both. We have cells that are both afferent and efferent of these cells. And if I click on this is a union ensemble. If I click on intersection, of course, the number of cells is very reduced, because intersection gives me the cells that are both at the same time afferent and efferent of this neuron that I've chosen here. Okay, can reset filter. Again, afferent, this one, and I can select 2 more neurons. Okay, now we are ready for the simulation. I will click on this. This will take a few seconds. And my circuit has been created here. Okay, so here we have, as you can see, the dendrites, the axons, and the somas of the neurons that I've chosen. And again, I can zoom in and zoom out. As we have seen the other web applications, we can click on individual compartments. We can click on the soma, and so on. This functionality is given because we need to decide the compartment from which we want to record the activity and the compartments or the compartment to which we want to deliver the stimulus. So let's say that in order to show some results, we will choose the same compartment. So I will take the soma of this neuron here, and I will tell the system that I want to record from this soma, and I also want to stimulate to this soma. Okay. In this case, a step current stimulation is added. The duration is we can set to 300 milliseconds, and again we can put 0.9 nanoampere. We can record also this soma recording, and also this soma recording. But of course, I mean, I'm just choosing almost at random here. If there's no connection between these two cells, direct or indirect connection, probably we won't see any activity on these ones. But I would like to show you the functionality and that this web application offers. So the fact that you can record from many compartments depending on your needs. So now everything is ready. We have a recording side, we have a simulation side. I can run the simulation. It's initializing the simulator with the parameters that we have given. Usually takes some time. And now the client side, which means this web application, this front side that you are visualizing on your browser, is talking to the server where the simulation are run. And you can see, as you can see, we have the activity of all the three sites that we have chosen for recording. As I was expecting, these two somas are not really generating any spiking activity. There's a subthreshold activity here to this neuron, because probably it is receiving some input which is not enough to fire an action potential. This neuron is not receiving any stimulation basically, and this is the soma, so the neuron that we have chosen for recordings and for stimulation as well. So you see that the look and feel is very similar to the single cell simulation environment. Also in this case, you can download the results here. So again, a CSV file is downloaded for results, and basically you can perform further analysis on the activity itself. Finally, very briefly, before we give you some time to experiment with all this web application, I would like to show you the last one, which is the brain area circuit in silico experiment, again with the hippocampus. Now, here you cannot choose by hand individual cells, but you can define a population. This will give you, as the name says, the entire region simulation. For example, I can decide to I don't know, take a slice here, or take a region, or take all the excitatory cells, for example. Then the simulation panel is opened, and I can edit the stimulus here. Yeah, for example, this one, I can edit the stimulus, the pattern, the Poisson, noise, population, the directional stimulus, and so on. I can choose what I want to report. In this case, the voltage of all the excitatory cells, which is the same that I have chosen to simulate. And then I can continue here with the simulation. Also, in this case, we can choose the service account. So this web application, again, is run on HPC systems, and is run through the service account service that I showed you before. Once the simulation is done, again, you can fetch the report and view the simulation if you have any. I haven't submitted any, so I don't have any reports here, but if I had a report, or if I had run some simulation, I will have a list of jobs that show me the simulation that are completed. Okay, I think this is what I wanted to tell you about web applications. I remember you that we have seen the feature extraction in the trace analysis group, and we have seen the single cell in Cdip experiment for the single cell experiment. Remember, if you go through this web application to select models that have the 2017 or the 2018 string in their name, we have seen the Hodgkin axon neuron builder, but we will not have time to go through the entire workflow probably. So if you want to play around, go through either the feature extraction or the single cell or the small circuit in Cdip experiment, the rat hippocampus CA1, in order to perform a simulation as the one that we have just seen. We will be happy to answer any questions if there are any, or just please take advantage of this last 10-15 minutes to play around with the applications and let us know if you have any trouble or if you want to know more. Thank you for your attention.